Hi, Jordan. So in our weekly journal club, I'm going to present this paper, Collective ERK AKT Activity Rate Orchestrate Epithelial Homeostasis by Driving Apoptosis-Induced Survival. So this is by a group of the researchers from University of Bern, Switzerland, and there are two outsider researchers from US. So maybe I briefly introduce the authors. So the corresponding author is Prof. Dr. Olivier Pertz. So he got his PhD in University of Basel, which is in Switzerland, and then worked as a postdoc in US. Further working in US for a while, then come back to Switzerland. So his specialist is more to the single cell analysis and cell development, cell morphology study like that. So he got a group of the members, including postdoc and PhD students, that kind of contribute most of the things in this paper. Paolo is one of the postdoc in his lab, and the major, I think the skill is about molecular medicine, and there's other co-authors that contributing to the mathematic model, the DNA model, and also the other web lab experiment. Then the two researchers from US, they are more working on the analysis and computational image sequence analysis. Okay, so come back to this paper. The overall idea of the paper is that they want to know, for example, the epithelial cell is we always able to maintain the cell integrity. They always have a constant number of the new cell versus dead cell. So how they actually able to maintain the cell homeostasis? This is what they want to know. But so from here they able to find that apoptotic cells able to communicate to the surrounding neighboring cells by releasing some signaling factors. So from there, the neighboring cell able to catch it and to regulate themselves and lead to the survival. So this is like a summary figures of how they actually have found in this paper. Okay, so maybe let's talk a bit about research background. The epithelial cells are frequently undergoing apoptosis, but then people always wonder why there's no such thing like the cell death in our skin layer, epithelial layer, so often. We always able to maintain it because the cell always have a good balance of the cell division versus cell death. So this kind of the epithelial homeostasis is depends on certain factors like the cell density sensing, epithelial exclusion, and the other thing. So this is what we have known from the previous study, and also a study that observed the Drosophila rings observed that the metagenic factor produced by apoptotic cell able to induce proliferation in neighboring cell, which able to enable wound repairing at the neighboring cell, but also some of them induce further apoptosis, and some other maybe trigger survival fate in also the surrounding neighboring cells. So this means that there must be a coordination of signaling pathway that lead to different cell fate, like maybe proliferation, survival, or apoptosis fate. So how it actually happen at the single cell level and special temporal manner. So what we already know about the pathway to trigger this kind of the coordination is like we already know these three signaling networks that must be involving. So MAPK is mitogen activated protein kinase or extracellular signal regulated kinase ERK, and phosphorinositide 3 kinase PI3K, or we can call it AKT signaling network. So this three obviously is important for regulation of cell fate. And now they also previous study also further identified that ERK or AKT signaling can fine-tune the phase decision at a single cell level. So this is why they say so, is because in the epithelial cell that discovered previously, ERK activity pulse frequency are kind of like different at the single cell level. And this kind of the differences, it depends on the epidermal growth factor concentration, and their activity pulse also is very correlated with the efficiency of cell cycle entry. So this is the same as the AKT pulse, and they are kind of synchronous. So maybe this means that these two factors maintain the metabolite stability in epithelia. Okay, so come to this result. So they kind of break the result finding in each part. So it's important findings. So the first thing is that they want to confirm that ERK, it's like the typo here is ERK or AKT activity wave. It actually happened in the epithelial cell. So they need to confirm this. So they don't really do the single cell analysis, but they try to use the imaging system, imaging analysis, that in a way that as seen by these figures here. So like as seen from A here, they try to transduce the cell with the factor, the factor that, I mean, the ERK, AKT, and the other H2B is the histone 2 factor, the biosensor into the cell, so they can observe it in real time. And so it's like after induce, immediately they can try to see the activity of this biosensor using the machine. So from here we can see after they able to generate it, we can see the differences in the signal wavelength, the what is that, the amplitude from time to time. So this is still very raw. They after they extract this, so the obviously the blue color and the green color is what they want to observe, and the red color here, the histone 2, is just for the control purpose, means that all the time they will just show similar expression. So as soon as C, they will try to process it and try to extract something so-called the ratio of cytosolic was over nuclear intensity. So this is representing the ERK or AKT activity. Yeah, okay. So so after they have set up these things, now is the time for them to test whether the ERK AKT activity affected by the EGF, the epithelial growth factor, or not. So obviously from the A here, without EGF or with EGF, they are like kind of the difference. And from here, when we know the signaling, we can also easily translate it into, convert it into the heat map, and for us to visualize it, like immediately. So from here, we also can see that the signaling, the ERK activity, the ERK or AKT activity, is kind of spreading in a like a waveform, in the radial waveform. So it's spreading to the surrounding, like how the water spread. Okay, so yeah, so they further to test that further to focus and to spot to target the cell and also the surrounding the neighboring cell in the different layer. So as seen from E here, E is the most obvious one. So let's say they already target one cell that is undergoing apoptosis. They try to target it and to observe it from time to time, so within the range of time. So from here, they can see that the signaling of the ERK or AKT activity wave is kind of the most strongest, most intense at the first layer of the label, and it gradually spread and in a lesser intensity into second layer and third layer. So this is the same as the other, they just can present it in other way like heat map or the other, the graph. Yeah, so this is like something they prove it, they're able to see it using this experiment setting. Okay, so next they try to also observe in other cell line. Okay, so just now the cell line is called MCF10A. So this cell line is the memory epithelial cell. And then they also try to test it in the kidney epithelial cell or maybe renal epithelial cell. So they actually found a similar observation as seen in the previous one. Yeah, so let's say to exclude that the chance that the waves are artifact, they also try to evaluate 3D the MCF10A as culture. From here, they're also able to see from 3D. So in 3D way, they also detect the similar activity of the ERK wave, which means that they started from the apoptosis cell. From 3D, we can detect it, and then also spread to the surrounding neighboring cell in the same manner. So all these results show that at least this ERK wave occur in different epithelial cell type, species, or cultural dimensionalities. So next, they want to detect what kind of the early morphological event of apoptosis is able to detect, to measure further. Means that the apoptosis, normally we have a lot of we have some of the signal or the sign to show that the cell is going to go through apoptosis. So they have the A to G here. They are actually different type of the event that might be happen before the apoptosis happen. So in this this cell line, they actually found that the most easiest way to observe is through the first one, nuclear shrinkage. So
is that what the tick and the cross are supposed to mean?
What is the what?
There's a tick and a cross on each of the
Oh
image.
yeah, the tick is means that they actually tested to see which event might actually let them to let them to track how when is the time the cell undergo apoptosis in this cell line. But eventually they found that the most easiest one, that is the nuclear shrinkage, because they're able to calculate the nuclear area in real time. But the other things might be not really very not really obvious in real time, so it's very hard to detect automatically. So that's why they try to just focus on nuclear shrinkage. So for the following downstream analysis, what they mean by apoptosis, they actually is a way like to detect the nuclear shrinkage activity. Is it okay? Yeah, okay. So after that, so this is like after they have studied, they try to check that whether this when after they test it, they just try to induce the apoptosis using inducer, a type of inducer is called octopax, that able to induce it within the single cell resolution, which means that they are only able to induce one single cell and for us to observe. So when they expose, when you put this into the cell and then they induce with the blue light, we can observe the apoptosis activity at the particular area. So this is what they want. And okay. So they want to see something like the mitochondrial outer membrane permeability, which means that the cell is undergoing apoptosis, and from there they can try to focus and to check the activity of the surrounding cell. Okay, so they actually try to test two type of the inducer, so orthobax or the MK. But the bottom one is like the caspase inhibitor is not really working, so which means that this caspase catch activity is not really influence the ERK AKT wave. Okay, next they try to also check that in, let's say now you know that ERK or AKT signaling pathway is obviously affecting the activity of the surrounding neighbor, right? So they want to know in this pathway what are the factors, what are the genes that corresponding to this kind of the activity. So that's why they did a lot of the inhibition. So each of the inhibitor actually inhibit certain particular gene in this ERK AKT signaling pathway. So from here they know that okay, so MEK is actually to inhibit ERK the ERK gene signaling, and then it sent to the AKT, and from here the result actually show that these two, the first two are mutually exclusive to each other, which means that the MEK able to inhibit the ERK activity but not the AKT activity. Okay, so
can you talk about what does the color represent? Is it a higher expression or is it lower expression?
Yeah, so color means that the blue are like no activity of these two things like ERK or AKT, and the and close to the yellow color means that this activity strongly strongly happen in that area. Okay, so blue is no activity, yellow or orange are a lot more activity. So this is the fluorescent color that happen at that area. Okay, so so other things that they found is that the EFFR signaling is obviously doing something to these two activities, because once they inhibit this EFFR signaling factors, these are no activity at all in these two in the area in the cell. So they did also the same in the starvation-induced apoptosis and also in the apoptosis-induced apoptosis. So which means that in the condition of starvation or in the condition of other external damage, this signaling pathway might actually the same. Okay, so they also just try to see whether is it just because the EGRF ligands, that wherever there is the presence of this EGRF ligand, whenever there might be the activity of the ERK or AKT activity. But so they try to compare two things. So one is that they constantly wash away the ligands, or without the water flow, means that the ligand is cumulatively building at the cell there. So with no flow, there is the activity there. So which means that the cumulative the accumulation of the EGFR ligand is no effect on the size or symmetric of wave. Okay, so next they want to know, this is something they're interested to know, is that the ADAM17 previous studies show that this involved in generating ERK signaling wave in vitro. So they want to test this also in this single cell level, and the result actually show that so with the in the wild type versus the ADAM17 mutation, they kind of just maybe delay a bit the activity of the ERK or AKT wave, and also maybe change of the size like that. But so means that it indicates that ADAM17 is partially regulates ERK or AKT wave. Yeah, that's all like that. Okay, so next they want to know this ERK AKT wave, what maybe other related with the cell exclusion. So cell exclusion is something like actually, let's say three things they put together. Let's say cell undergo exclusion means that they will just push away the apoptosis cell outside. And yeah, these are it's kind of like, what is that called? Yeah, I don't know how to show it.
They push the dead cell out of the system.
Yeah, out of
so
them.
They push it outside. Basically.
Yeah, push, yeah, something like that. So they just use the image to check the monolayer of the cell, the floating cells, to see whether it happen or not. So and then they try to induce different type of the inhibitor of the ERK or AKT signaling pathway. But they found that this is not really something very not correlated with the cell exclusion, which means that without the signaling factor, the cell exclusion still happen there. Okay, so next they try to see, they try to use the convolutional neural network to predict the activity of, like, whether to predict the fate of the cell, whether it will become apoptosis or it will just non-apoptotic.
Yeah, you mean convolutional neural network, right? CNN.
Yeah, convolutional neural network.
Yeah,
just want to predict the fate of the cell based on the ERK or AKT wave. Yeah, so they try to generate a lot of the data by comparing, like the first one here, comparing the ERK trajectories before apoptosis versus non-apoptotic cells in 6 hours. And within this 6 hours, the image is kind of captured around every 10 minutes, yeah, if I'm not mistaken. Then they use this data to train the CNN to classify the cell fate. Yeah, so A, B is kind of like the activity that after the train, they kind of like already can group them into 2 different categories. And C is like they kind of like get the distribution of the ERK activity as predicted by the CNN output. So they use this CNN output to try to predict the collective ERK activity event. So from here, they not observe that in apoptosis cell, the cumulative event is how to say, there is with lesser of the cumulative event of ERK or AKT wave. While in the non-apoptotic cell, they actually having more ERK AKT wave. So which means that when the cell experience collective ERK event, they are less likely to die than the cell that do not. Okay, so next they also try to check is there any local survival signal, which means that they try to see the neighbor surrounding the first layer neighbor, second layer neighbor surrounding the apoptosis cell, to see that while this ERK AKT wave happen, how long they can able to sustain to make them survive before they go to apoptosis also. So the result show that up to 4 hours is significantly less likely to die, which means that once the apoptotic cells inform the surrounding, the neighboring cell that inform them with the ERK AKT wave, they are like up to 4 hours, they will just keep surviving before they gradually maybe undergo apoptosis together. Okay, so next they want to try to check the survival rate as a population. Yeah, so they try to use 2 optogenetic system to evoke different signaling pulse frequency and to observe. So they try to check the pulse pulse frequency in different timeline, and from here they actually just found that with the reduction of the apoptosis rate with triggered ERK pulse at higher simulation frequency. So which means that let's say we induce more and more ERK pulse, the cell is more likely to survive. So they actually term this something like the So okay, so next this is the last result here. They kind of like already can confirm that this type of the signaling is able to drive the neighboring cell to survive longer. So they term it as the name as AIS. So apoptosis-induced survival. So this AIS maintain epithelial integrity in response of environmental insult of different intensity. So how they actually do this is that they try to show three scenarios. Like they try to do three scenarios here. So like as seen as the C here, scenario one is like with high activity of the production, which means high activity of the ERK wave, and also with low apoptosis rate. And the second one is the high protection and high apoptosis, and the third one is the low protection and the low and a high apoptosis rate. So they try to compare these three scenarios to see what will happen in the neighboring cell. So as seen from D here, which is the most easiest to understand. So in the first scenario, which is the normal apoptosis event would happen, the surrounding cell is still maintain the cell count, which means in the population level. And as let's say we just try to increase the apoptosis rate, but also with high production rate. So the cell is still able to maintain. So which means that the ERK signaling is really something doing on the neighboring cell to make them survive. But let's say in third scenario, which means we increase the apoptosis rate, but we reduce production of the ERK wave, it's obviously that the cell count will drop after certain hours. Yeah, so from here they kind of like further confirm that this ERK wave is something to do with the survival rate of the neighboring cell. Yeah, so this is also something else they try to measure the instead of the inside just now, this is that they measure the hole formation. So the hole formation, they also use certain mathematical model to detect the hole within the image, the real-time image of the cell there. Okay, so from here again, as seen from Jacques here, within these three scenarios, I mean the output of these three scenario is similar with the previous one. Okay, so in summary, this apoptosis-induced survival signaling mechanism was found that to preserve epithelial homeostasis in response to environmental stress. So at the single cell layer, at the single cell scale, AIS involved apoptosis trigger ERK activity wave, and at the population scale, AIS allow the cell collectively to adjust survival rates to make them more able to survive. So the thing is that so this is their finding, and the limitation of the paper is just that they only focus on how apoptosis-induced ERK activity wave regulates survival, but they don't really measure also other signaling pathways that might be doing the same thing as the ERK or activity wave. Yeah, so this is their limitation. So that's all for my sharing today. So thanks for listening. Yeah, so now maybe open like to let you to ask me some questions.
Yeah, I just want to make sure a few things because I think I have asked all the essential questions during the presentation just now. So it's beautiful. The presentation is very clear, and I kind of understand what they're trying to do, especially with the summary in the end. But first of all, what is a mitogen? Mitogen, I think, yeah.
Yeah, mitogen is like cell division, the stage.
Yeah.
Yeah, they got the three stage. Mitogen is like meiosis, mitosis.
So mitogen should be the agent to induce mitosis.
Yeah, induce the cell division.
Okay, yeah. That makes sense. So mitogen inducing agent. So what do you mean? So in one of the slides you have mentioned something called pulses. So how is the pulses measured in the original paper up there?
Yeah. So
do they talk about it?
This is what I think it's the abnormality, sharp abnormality.
Yeah.
So the so-called pulses is like a cumulative signaling intensity of the image. Yeah, so this is the biosensor. So they constantly release the color, but so means that over the time they measure the intensity and convert it into the signal. So from here they can see that the signal is come in a way like sometimes they suddenly go up and then gradually go down. It's just like a pulse like that, just like a pulse form. Yeah, something like this.
Okay, that makes sense. How about there's a word called quiescent. What does quiescent mean? Yeah, that's up there. Apoptotic cell in quiescent unstimulated epithelium. What does that word mean actually?
Oh yeah, I don't know. So yeah, what do you think?
Okay, doesn't matter. We'll just put in the comments or put in the video description.
Yeah.
I don't know. That's why I'm asking the question.
Yeah.
So just to summarize the whole paper, they found that the apoptosis in the skin cell is actually part of the very essential pathway to maintain the structure and integrity of the skin cell so that your skin cell doesn't develop into cancer. Okay, so how do they do that? It's actually through the ERK pathway or AKT pathway.
Correct? I think not lead them to become cancer. I think it not lead them to to die so early.
Okay, that makes sense as well.
Yeah, it's just a normal cell in the healthy people cell. Yeah, so let's say one apoptosis happen, how actually the surrounding cell not die together. Yeah, so this is like because they have this kind of actually this is something like ERK wave. Yeah, that happen. Yeah, to save them for a while.
So in this case, they tell the neighborhood cell not to die at the same time.
Yeah, not to die. Yeah.
Okay, that's very interesting. That's good. That's a very interesting.
Yeah, and actually, I mean, this is really something that they're able to visualize it in this type of experiments. I think it's just that previously very hard to detect it. Previously, people only do in vitro and then maybe detect the signaling activity using some post analysis, not really the real time. Yeah, so this is something that very good for.
I mean, this is not part of the paper, but do they talk about the future direction of this pathway? What we could use them in the future studies, or how can we use them in applications and so on? Is there anything in the paper to talk about those?
I believe that they talk a bit in the discussion. It's just I didn't highlight those part. Yeah.
Okay.
Maybe this is, but I mean, fundamentally, this is something just to understand deeper how the cell development happen like that.
Yeah, makes sense. Okay, thanks. That's all the questions that I have.
Yeah. So thanks for your time. I think today we used around 30 minutes. Half an hour. Yeah.
Okay, so thanks.
Okay, so thanks.
Bye.
Bye.